Alright. Guys, so today we are going to have a bit of a very chill, relaxed activity. Okay, so the first two classes were just an introductory session for you guys to get to know various tools, and we had already mentioned that we'll be exposing you to how they are being applied in real life, right? So today it will be a start to that. Okay. So from since we are starting from the basics, we'll go with small problem statements, or like small case studies or scenarios, and then we will take it to the next level. Okay. So for now, it's going to be a very small case study which we are going to be dealing with today. So I sort of expect you guys to be a bit more interactive today. If you can't unmute also, at least like give a thumbs up or show some reactions. And I do expect you guys to give something in the chat box also. Okay. Let's keep it a bit interactive, guys. So even if you can't unmute, at least put it up in the chat box. Is that okay? All right. Thank you so much, guys. Okay. So, do you guys remember what happened in our first session? So, we were introduced to four tools, right? So, can anyone summarize on what the four tools were? What all we used? Anyone, guys? You can put it in the chat box also. Can anyone tell me what are the four tools we used in the first session? Okay. Were you guys there for the first session or did anyone miss it? If anyone missed it, can you give a thumbs up here, please? Alright, so all of you guys were there, right? Okay, so we'll start. We'll just start with a very small summary of what happened in our first session. Okay, so the first tool we got introduced to was NCBI. Can anyone tell me what NCBI is? Like what we use it for? Guys, be a bit interactive. Yeah. Yes, exactly. That's right. So, Shani, yeah, you're absolutely right. So, we got introduced to these four tools, which was NCBI, Uniprot, TDB, and SOPMA. Amazing, great. So, can any of you tell me what NCBI is?
So NCBI, we learned it has genomic information and stuff,
Hmm.
and it also has multiple databases like BLAST, GenBank.
Yeah, exactly. Yeah, that's great. Mantra, amazing. Good job. So, what she said was absolutely right. So, Uniprot is a collective database for biotechnological information. You have various set of informations, including your genome sequence, protein sequence. You can blast. You can also get a lot of papers, review papers, research papers, where some are in open access, and that is where what you get under PubMed Central. So that was PNC. So all of these come under NCBI. So mainly, if you want to get a nucleotide or genome sequences, you go to NCBI, right? So I mean, good job. Now the next one which was mentioned is Uniprot. Can someone tell me what Uniprot is?
It is a database for protein sequences.
Yeah, exactly. Yeah, that's great. So Uniprot is a database where you will be analyzing all your protein sequences. So can anyone tell me what is PDB and the difference between what Uniprot and PDB? Yeah, Ananya, go ahead.
From what I understood, Uniprot was mostly for looking into the sequences, and PDB is more about the 3D structure of the protein.
Exactly. Yeah, you're right. That's perfect. So that's exactly as what she said. Unicode is only for you to analyze your protein sequence. But PDB actually gives you the three-dimensional structure of the protein based on some X-ray crystallographic studies, or it can also be from computational data. But it will give you how the structure of the protein might look, whereas your Uniprot gives you the protein sequence, a set of amino acids which make up the protein. Okay, so this is the main difference between Uniprot and BDD. And finally, SOPMA. Can anyone tell me what we use SOPMA for? Exactly, yeah, Mantra, you're absolutely right. So it gives us the information on secondary structure of protein. So these four web tools are very basic. Okay, so we'll be using these. pretty often, especially when it comes to Uniprot, PDB, like I mentioned the other day itself. So if you want to work with a protein, you would require its PDB format and obviously get it from your PDB website, right? So these are very basic tools which we'll be using very commonly, okay? So today we'll be starting with a very basic case study, but before I move into the case study, I want to introduce you guys into another web tool which is part of NCBI. Okay, so we go into NCBI, and I had already mentioned about this before, but we didn't go much into it. But this is called the ClinBar website. Sorry, just once again, I'll share the screen to you guys. Let me know if you can see it. Can you guys see my screen? All right, that's great. So we are going into NCBA, and under this, we go into the TrimBar database. Okay. Um, just for now, I'm going to give like a random protein because I just want to introduce you guys into what ClinVar is. So we'll just take insulin. Just something random because it goes into the page where you can see about what it is. Okay. Yeah, so you go to the about here, you get the introduction to what this ClinVar website actually is about, database actually is about. So as you can see here, it is mainly to give you information uh, about different diseases and pathogens, different variants that can happen in your body or in some proteins, which is responsible for causing a disease. Okay, so for example, you have some mutation in your gene sequence. So this ClinVar website, it has data curated from multiple web pages, FDA approvals, etc. All of these are approved data. So it will give us information on whether this particular mutation in your gene can actually cause a disease or not. It can also tell you about how effective drugs are. For example, uh, if you are taking a drug for quite a while, when generally it has a effect to say lower your blood glucose levels, you don't see the appropriate results. So what could be the reason? Just one second. Yes, sorry. So we just want to know what is preventing the drug from showing its effect. So from this ClinVar database, we can see about how the drug's effect might be different if you have a variation in your gene sequence. Okay, so ClinVar will basically tell you a lot of information about uh, diseases and how your body is responding to it, or what diseases you might acquire. Dave, I'm sorry, can you just give me a minute? I'll be back. Yeah, sorry guys, I'm not feeling a bit well. That's why I had to take a break in between. I'm sorry. So yeah, we can continue. So as I mentioned before, yeah, ClinVar is just going to be a database for you guys to know what a disease is going to be caused by any variation in your gene sequence or protein, and it also gives you information on how a drug has an effect on your body. Okay, so this is what ClinVar database is about. Today, we are going to be seeing a small case study in which we'll be using this web tool. Okay, so now this is your case study. Okay, so they are saying a 42-year-old female patient undergoes tumor genome sequencing. So this patient has a tumor. We are taking a part of the tumor and sequencing its genome. So before we move ahead, can someone tell me what do you mean exactly by sequencing a genome? Or can someone tell me what a genome is? Anyone? If you guys don't know the answer, you can give a thumbs down also. Yes, no, the that is a different thing. See, it's almost there, okay, but it's not exactly what it is. Sequencing a genome, actually, what it means is to know the entire sequence of genetic material that makes up a particular tumor. Okay, so from that, we'll be using it to, compare it with two different reference sequence or a normal sequence. So we will use an entire genome, and then we'll be comparing it with a reference, and that is called BLAST. That is called an alignment. But in general, sequencing a genome means finding the entire genome sequence, that is the set of genes which make up that tumor or that make up that particular cell or tissue. Okay, so that is what genome sequencing means. Usually, we go for genome sequencing when you want to go for mutation studies, etc. Okay, so now here they're saying this person has a tumor and they are doing a genome sequencing, and they have identified, and this person is from a family who has a history of early onset cancers. Now they have identified a particular mutation in the TP53 gene. Okay, so now actually, what this means is that there has been a mutation, there has been a change in a particular base in the TP53 gene. What the TP53 gene actually codes for is the P53 protein. Does anyone here have any idea about what P53 is? If you don't, again, give me a thumbs down, but please be a bit more interactive. So, does anyone know what P53 protein is? You can give me a thumbs down, guys, if you're not aware of it. It's fine. All right, no issues. So, so basically, what happens in our body, the tumor suppressing mechanism, right? So, what happens is there are some certain proteins in our body which, so I'm sure you guys are aware that all of our bodies have cancer cells, but all of these are silent and they're kept under control. Our immune system has something called tumor suppression mechanism, where if any cells start multiplying rapidly uncontrollably, there will be a mechanism to make sure that this doesn't spread anymore. So one such mechanism, one such protein which takes care of that mechanism is known as P53. Okay, it ensures that once cells attain a specific age, after that they have to die or undergo senescence. If they keep on proliferating after that, that's what means we have cancer. Okay. Yes, exactly. Yeah, Urjusudri, you're absolutely right. So P53 triggers apoptosis. Exactly. So as I mentioned, it should not allow your cells to proliferate any further. It needs to arrest it. Okay. So once your cells age, it will call the senescence of the cell. Senescence means death of the cell by causing apoptosis. Okay. So this is what normally your P53 gene does, P53 protein does. So it is a tumor suppressor protein. Now what they are saying is in this protein there has been a variation and it is denoted by this. Okay, so you may ask what this actually means. So here, this C here means the coding sequence. You remember in NCBI we saw about the CDS. CDS means the coding part of the sequence, right? So in the coding sequence, at position 743 of the entire gene, there has been a base change from G to A. I hope you guys have understood what this notation stands for. In the coding sequence, at position 743, there has been a change from base G to A. Okay, so this is what it means. Uh, do you guys understand? Is it going fine, or am I being fast, or is it not clear? All right, that's great. So now, yeah, as I mentioned, there has been a base change from G to A. So this is one notation. Another notation, what they're saying is, at in the protein level, there has been a change at position number 248 from arginine to glycine. There has been a change in the amino acid. Okay, at position 248. So this is how you will actually represent this. Okay, now, this part can actually be represented with their amino acid sequences also. You know how, yeah, like you say, arginine and glutamine, there has been a change, right? So, arginine is represented by an R, then glutamine is an amino acid represented by Q. These are like single letter representations of your amino acids. So, here you can again represent this as P It changes from arginine at position 248 to glutamine, which is Q. Okay, so this is the notation to show that there has been a base change at that particular position in the protein level of this protein, while at the genetic level in the coding sequence there has been a base change from G to A in precision 743. Okay, so what our case study today is saying is that from the tumor genome sequencing, they have found that the P53 protein has undergone a mutation at the 248 amino acid from arginine to glutamine. So today, our task is to see if this mutation can be considered pathogenic, or if it can actually cause cancer, if it can alter the activity of p53. Because if p53 is altered, what could happen? There could be no apoptosis. It will not have a control on all your tumors, the highly proliferating cells. Then that is what leads to cancer finally. Okay, so you need to know if this tumor is cancerous or not. uh, because of this person has a mutation in the P53 protein. Okay, so that is what we are going to have a look at today. Did everyone understand what the problem statement is about, like what the case study is about? Is this clear? All right, guys, that's great. So let's start now. Now, I want you guys to give me some inputs on how we are going to be going about this process. Okay, you tell me what you think is right. So we will not be. I mean, there's nothing right or wrong here. You guys can just go about saying what you think should be right. Okay. So first, I need to know what the P53 protein sequence actually is. Okay. So from then only I can know where the base changes, and then I can identify what all problems are there. So initially, which web tool do you think I should use? Anyone?
Uniprot?
Exactly. Yeah, you're absolutely right. So we are going to be using Uniprot only. Good job, Ananya. So now, so as Ananya mentioned, we will now be going to Uniprot. Okay. So what protein we are looking for is P53, right? So we will be just entering the P53 protein here. Now, if you guys had noticed in the last class, I would have told you one very important thing to look at. Can anyone tell me what it is? So it is basically the next step from here. Can anyone tell me what it is? Like you
To
can tell
click
me
on
if
human.
yeah, exactly. You're absolutely right. So we need to go to specifically humans so that we can get the sequence only in the required organism, which is human. Because in our problem statement, we have a 42-year-old female patient. Okay, so you go to human, and then under that you go to your first entry because that is what is the Uniprot ID for cellular tumor antigen P53. And here also you have the gene name TP53, and that is what is mentioned here also. Okay. So now you go in. So you have your gene name, which is TP53. And it's present in Homo sapiens, which is human. It is called cellular tumor antigen P53. And it has 393 amino acids. Now, can anybody tell me what this protein existence means and what this annotation score here means? Anyone, guys, if you remember from the previous class, what I mentioned? If you don't know again, it's okay. It's fine. If you don't remember, you can just give me a thumbs down. I can brush you through on that again. Yes, go ahead, Anand.
Yeah, so protein existence means that we have actually observed it because some have only been predicted computationally.
Hmm.
And annotation score is how accurately we have sequenced it.
Yeah, your protein existence definition is absolutely right. Annotation, you're almost there. So, okay, yeah, Sai, Aishwarya, you may go ahead.
So if the annotation score is high, that means that the functions of protein is well known.
Exactly, yeah, you're absolutely right. Ananya, you did an absolutely great job, and yes, Aishwarya, that's perfectly right. So, protein existence means that the protein has been identified at its protein level only, and annotation score it has a high score means or most of its function has been mapped. So, we know the function of this protein. Okay, so now what we need to know is there has been a base change. So you can go to the variant viewer and look at the variations, but you can see how many there are, like 3,163 variations which have been identified. Under this, if you need to go look at this, so let's say you are able to go identify base number 248, right? Was it 248? Yeah. So at 248, R changes to Q. So now, where was yeah, wait. So if you zoom in further, so here you are at 240. So 41, 42, 43, 44, 45, 46, 47, 48. So at this position, at position 48, you need to see where the base change is occurring from R to Q. So you can see here, Q is somewhere over here, right? This is only Q. And then if you click this, you will get a data, and I think the server is also now running slow because there's a lot of data for you to look at. So in this case, it will be very difficult for you with when you have so many predictions here to look in the variant viewer to see what might be the possible effect of this variation. So instead, what we are going to do today is we'll go into the feature viewer. Okay, again, we have like a large number of features, but this has been represented in a much more easy to understand manner. So you'll be able to understand what is actually being mentioned here. Okay. So now you have a lot of these over here, but we don't need all of them. We just need, say, for example, sites. Okay. So now you can we are going to assume the situation to be that there has been some mutation in the p53 protein, that it is not able to bind to DNA anymore. If it cannot bind to DNA, then it will not be able to stop the replication and cell proliferation, right? So we will see if this particular mutation, that is the change from R to Q at position 248, lies within the DNA binding domain or not. Okay, do you guys understand what I meant? Is it clear so far? Do you guys understand like why we are be going into the DNA binding? Okay, that's great, guys. So, like I mentioned, we'll be seeing if this mutation is within this DNA binding site, that will cause the protein to not bind to DNA anymore. Okay. So, as we saw, this DNA binding site, if you click on this, you will notice, you can see here under the sites you have a DNA binding site and just general binding sites also. In this DNA binding site is from position number 102 to 292. Okay, so from amino acids 102 to 292, all of these lie under the DNA binding domain. And our mutation is occurred at position 248. So this lies within the DNA binding domain, which means that we can conclude that yes, okay. So if this mutation is occurring within the DNA binding site, then yeah, there could be some effect on the p53's effect on tumor suppression, right? Do you guys get it? Am I clear so far? All right. So yeah, so that is what we are trying to conclude from Uniprot. So yes, now we have ensured that okay, there has been some mutation over here which is causing a problem. Next, we are going to see if this is actually going to be cancerous or not. Okay, sometimes it may not bind to DNA, but it may not necessarily be pathogenic. Okay, there are some situations like that also. So now this is when we'll be moving to our next database, which is ClinVar. Okay, but before we move from Uniprot, we always require one major information, and that is your FASTA sequence. Okay, so we will just get our FASTA sequence for this particular protein. We'll just have it ready. So if at all we'll be requiring it in our next steps only. So for that time we will be using it. So for now, let this be here. Okay. So next, we are going to identify whether this particular mutation, that is at position 248 from R to Q, is going to be pathogenic or cancerous or not. Okay. So now what we will do is we'll go to the ClinVar. I'm sure, I mean, from Uniprot, you can see that yes, it lies within the DNA domain, and you can confirm that okay, yes, it is going to be causing a problem. and tumor suppression. But you cannot directly decide that. You need multiple sources to say yes, it is a problem. Okay, so that is why we'll be verified with all these websites. Now we go to ClinVar. Okay, in this, so once you go into NCBI, you will choose ClinVar over here. Now, what you will do is, remember in Uniprot, we saw that the gene name over here is called TP53, right? Yeah. So the gene name is TP53. So you can just copy the gene name, paste it here, put a space, and just copy paste this notation to say what the change is. Okay. You will just paste it over here. Okay, so what we are saying here essentially is in the TP53 gene, there has been a change at this position. That is what we are trying to input into the database. Just click search and you will get the classifications here. That this is what we will get. Okay, so now from this data, what we can understand this. Okay, so here also it mentions that if there's a change at the protein level from arginine to glutamine. That means there has been a change in the coding sequence at position 743 from G to A. Okay, that's what this is saying. So here it will give you information about two parts. One is germline and one is somatic. Can any of you guys tell me what the difference between germline and somatic is? Anyone guys? What is germline and what is somatic? Yeah, yes, Sandeep, you can go ahead.
Yeah, germline means it is involved with gametes like for males it's the sperm and
Yes.
females it's the ova, and
Hmm.
somatic is the body cells like every other cell which
Exactly.
needs the tissue
Hmm.
paper.
Exactly. You're absolutely right. Okay. So that's exactly what germline and somatic mean. Germline means it is passed on through your sex chromosomes, while somatic means it's present in all other cells of your body. So here, what they mean by germline and somatic is that if this particular disease, like Clinvar by itself, is only going to show you, the information related to human diseases or health, right? So this particular disease, is it passed on through, your generations? Is it passed on through your family, or if it is just present in all your somatic cells? That is, it is not a genetic disorder, but you just acquire it, or it is due to some mutation which was present in your cells. So something of that sort, but it is not acquired through your genetics. So that is what your somatic mean. Okay, so that is the difference between germline and somatic. So now under the germline, so it has been reviewed by an expert panel to say that yes, this particular mutation is pathogenic. Okay, so whatever we saw this mutation here, they have classified it as pathogenic and it has been FDA recognized. So it has been confirmed that yes, this particular mutation is going to be pathogenic. Okay. So now, how exactly is it pathogenic? What exactly it is going to cause? You need to know that as well. So that is what it is. We have it. You will be given what it actually causes here, and here in this case, it is called as the Lee-Fromming syndrome. Okay, you can uh, open this to see what this syndrome actually means. Okay, here. So yeah, you have an entire data about what this Lee-Fromming syndrome is and what its symptoms are, etc., etc. So here to just know what this is. So LFS is actually a cancer predisposition syndrome. So it actually has a high risk associated with broad spectrum cancers, which includes five different kinds of cancer which has been majorly identified, including adenocortical, breast cancer, central nervous system, etc., etc. Okay, so this has been confirmed that this particular mutation will lead to cancer, and it is causing all of. I mean, it is causing the Lee-Family syndrome. It has been recognized and identified by the ClinVar database. Okay. Now, when you come to the somatic side of it, here they do not have much information regarding that, so there has been no data submitted, and only one out of the two submissions say it is oncogenic. So we cannot exactly say how whether it is directly caused by some mutation in the body, but we can for sure say that it has been passed through your genetics. Yeah, Ananya, go ahead.
Yeah, I had a small doubt.
Yes.
What exactly is the difference between pathogenic and oncogenic?
Okay, see, oncogenic is specific to cancers. Okay, this, like I mentioned before, ClinVar is a database which tells you about diseases in general. It is not going to classify whether this disease is related to cancer or that disease is not. It is not going to do that. So, in general, when you have a when you give a mutation to them, that when you say that this in this particular protein there has been a mutation from one base to another, what will this cause? And they are saying that is pathogenic, which means it will cause a disease. Pathogenic means disease. Okay, so oncogenic means cancer. So these are two different things. And under pathogenic you will be seeing what disease it exactly is. Okay, and that is what the Leaf-Fermini syndrome is. Okay, is it clarified?
Yeah, thank you.
Okay, cool. Yeah, Mantra, go ahead.
So what if we do not have an expert panel submission to some data, but still it is oncogenic or pathogenic?
See, sometimes yes, you may not get exactly the required data, and you won't be able to exactly rely upon it. But if that is your only source of evidence, then you have to take it without any choice. Like I mentioned before, not all your bioinformatics studies are valid because they may still be under research, or you have very few literature to support that fact, so it may not be reviewed. But if you are working on a particular research and you really need some evidence to support your fact, or you want to say build up further on it or study more about it, that is when you will go for all those data which are not yet reviewed. Okay, all right. Shall we go ahead, or is there any other questions, guys? All right, cool. Let's go ahead now. So from this CLIMA database, we have found that yes, this particular mutation is pathogenic. Okay. Uh, Now, if we want to say, okay, now I want to know what actually happens to the protein structure. Because let's say if I do know this happens to the protein structure, then I can try to find some other replacement or some other protein which resembles a similar structure, so that it can take over the role or something of that sort. Okay, it's not exactly that, but just I need to know what is happening to the protein foldings that is causing this particular protein to become completely dysfunctional. Like, is it being a major change or no? And even if it's a minor change, why is it causing it? Okay, I just want to know that fact. Again, you may have already decided at the uniplot step itself that yes, this is pathogenic or this is cancerous. This mutation is bad. You may have decided at the uniplot level itself, but you do need multiple stages of confirmation to say that yes, there has been some variation and this mutation is harmful. So that is why we'll be going through multiple of these steps. Okay, so next you have confirmed through CleanVar database. Now finally, I need to know if the folding of the protein in its secondary level stage is having any major changes or not. In that case, can anyone tell me what website we'll be using? PDB is only to analyze the existing protein structure, but I want to know what is happening to the folding. So yeah, exactly, that is SOPMA. Okay, SOPMA will give me information on how the foldings change. PDB, you can only visualize the existing P53 protein, but you cannot visualize its mutated form. Okay, and by the way, just to know, I mean, just to let you guys know, to get an X-ray crystallography study of a particular protein, it takes nearly 2 years for you to perform the analysis. So that is a very long time, and that is why you have multiple entries on PDB itself, which are computationally derived. Okay, so it takes very long time, and that is why your uh, this bioinformatic databases are being of more use these days because it can predict the structure much more quickly. Imagine having to wait for 2 years to see if this is structure or no, when in bioinformatics you can get it very quickly. It may or may not be right. That is the only issue we are facing today. but still there has been a higher rate of accuracies nowadays with all improving servers. So yeah, so hopefully in the future we'll be getting greater accuracy and lesser use of all these experimental data. Okay. So yeah, as we now thought of mentioned before, we'll be going to SOAP. So now I'll be opening two SOAP pages simultaneously over here because I need to compare what is the difference between the original P53 protein and the mutated P53 protein. Okay, so that is why I'll be opening two pages over here. So okay, this is the older, newer website. This is the one. Okay, this is the newer website. Let's go to this. Okay, so like I mentioned before, first we will be choosing this 4 helix, and these all are other default parameters, so we don't have to make any changes in these. Okay, let these remain the same. Now, here I need to paste the protein sequence. So from where should I get that?
I think you downloaded it earlier.
Exactly, right. So from Uniplot, we'll be taking our FASTA sequence, which we downloaded earlier. Okay, so you can download, and this is the FASTA sequence. So you will just be copying this protein sequence and pasting it in Sigma. Okay, and here after you have changed it to 4, the configuration states to 4, you can click submit. Now in the other page, what I told you was we'll be comparing it with the particular sequence change. So what we will do here is uh, we will be manually changing that particular sequence, okay, at the position 248. So under the sequence, you can just copy this one because this is what you have from 241 to 250, right? So we'll just be copying this, and I will just put find, paste the sequence. Now here, this is 250, 49 and 48. So in 248, I will be changing it to Q, from R to Q. So that is what our change was. So we'll be manually making this change. Alright. And again, this is 4 helix. You give submit. So now in this previous one, you can see what the percentage of alpha helix and beta sheets and all of that is. So you can see here that alpha helix is almost 18%. You have 71 alpha helices, and then random coil is at a highest of 64.12%, where they have about 252 random coils, then 16.79% is extended strands. Okay, so these are the major forms that you're taking, and you have a small percentage of, which is about 1% of beta tones also. So this is in your wild type. Wild type always means that it's the original form of the protein. Okay, so that is what your wild type means. Now, so yeah, if you now go to the other one where there has been a change in the sequence, the protein sequence. Here, what you can see, there has been a change in the percentage of alpha helix. So from 71, it has reduced to 67. Okay, and then again in your extended strand, here you had 66, and now this increased to 67. Okay, so it may or may not change, but our aim is to just understand how much variations are there. Okay, so here you can see you had 255 random coils in your mutated type, whereas in your original type you had only 252. So now if you actually compare the two, you will notice that there isn't much change. So since there isn't much change in your folding patterns, you would initially conclude that okay, if it is almost similar, then there shouldn't be a significant impact on the function of the protein. Because there has been a very minute change only in its secondary structure. But you do notice that there has been a major, I mean, it is actually pathogenic, and it does lie between in your DNA binding domain. So it is a very significant mutation, and it is cancerous. So we cannot just ignore it and go. So what I am trying to convey from this data is that you may not have significant changes in your protein structures, but that does not mean that the effect of the protein's function is also not going to have a significant variation. Okay, that is not at all correlated. That is why we look at every single data which we can find in a bioinformatic approach. Every data will give you new perspectives to look at. When we saw in the Uniprot, we knew it is in the DNA binding domain. When we saw in ClinVar, we knew it is pathogenic and almost partly oncogenic, because one paper says that it is oncogenic. Okay, and under the pathogenicity, you were able to see that what exactly it was causing, and in some germline cells, it did cause your Frommeni syndrome. Okay, so, but then when you went to your SOPMA, you only saw very minute difference in your secondary structure. So initially, if you had checked in SOPMA, you would have concluded that okay, maybe not. It is not causing that big of a change. Okay, so that is what. So this that's the end of this activity. So we'll be able to conclude here that yes, it is considered pathogenic. The structure may not significantly change a P53, but its effects and its function will be vastly affected by this particular mutation. Okay. So are we clear on this, guys? It is a very small and very simple activity. You can do this for any organism. For example, you have beta lactamase production from Escherichia coli, E. coli bacteria, and if there's a mutation in one of the uh, genes or one of the proteins coding for this enzyme, then will that be much of an effect on its function? So these are like very small cases where you will be using all these tools simultaneously, so that you will be able to have a better understanding of what the final effect is. Since we took for a very simple study, we were able to get good data for all of this, and we were able to have a good conclusion. But when you're taking new compounds, or when there has been new mutations and new proteins that are coming to existence, or we are only now discovering these, then in such cases it will be very difficult for us to judge with one or two websites. So that is when we'll be requiring a lot more tools to assess and see that how much difference that mutation is going to be causing. Okay. So are you guys all clear on on today's topics, on today's case study? Did you find this useful to see how you will actually apply the your databases which you learned the other day? All right, guys, that's great. Okay. Now, can someone like quickly summarize to me what we exactly did today? Someone who hasn't spoken before would be nice. Anyone?
Hi. So,
Yeah, yeah, go.
we basically considered a problem statement where we have someone who underwent a genome sequencing because
Hmm.
they believe that they were susceptible to cancers, an early onset
Yes.
cancer, and the
Hmm.
result of that sequencing resulted in mutation in this TP53 gene, which is important as a cancer marker, and
Yes.
we saw that we which exact position there was a change in. So
Hmm.
we learned to read the format in which we're going to be given these problem statements. We'll encounter them
Hmm.
that
Okay.
in the and then after that we considered the
Hmm.
position that it's changed from arginine to
Hmm.
glutamine at this particular point in the protein sequence.
Hmm.
So we go to again, as I'm not this is my first session, so I'm not particular about the website names, but we
Okay.
go to our first website, wherein we take the we download our Uniprot. So we download
Uniprot.
our
Yes.
p the uh,
The
like the
protein
sequence,
sequence. Yeah.
protein sequence that we want, and we keep that ready. And then also
Hmm.
here we can check if the it's pathogenic or not.
Hmm.
Like if it's a
Hmm.
very popular like something we're analyzing is very popularly
Um,
studied, then we can immediately at this
hmm.
point in our search
Hmm.
analyze if it's going to be pathogenic. And there are we
Hmm.
learn the different tools where we can consider our like different viewpoints of considering the sequence we're working with. And then
All
after
right.
this
Yes, yes, go ahead.
uh, yeah. So then after that, we move on to our next website where
Hmm.
the NCBI website where we use ClinVar, which is
Hmm.
a database, and we put in our gene and then we put in the variation that we're considering. And we
Yeah.
received two analyses, a germline analysis and a somatic analysis.
Hmm.
The somatic one show that it could be a possible oncogenicity in the variation we're considering,
Hmm.
and the germline showed us that there is a established syndrome that
Okay.
is associated with this variation, and
Hmm.
gave us a certain
Yeah.
amount of
Okay.
information.
Yeah. Yes. 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 You're absolutely right. So, does anyone else want to take over? Because you have done an amazing job. You have done for both tools together. Does anyone else want to take over? Good job, David. By the way, amazing. Anyone else, guys? You can take over from wherever she stopped. Anyone else who wants to just give it a try? Yes, Akrit, you can go ahead. Yes.
Yeah, so after you check the
Hmm.
pathogenicity in ClinVar, you then copy the amino acid sequence that you got from the FASTA file and you go
Hmm.
to SMOKMA and you basically copy paste the sequence, then original
Yes.
sequence without the mutation.
Hmm.
And then you open another tab and do the same thing, but at the 240th location, you change the R to
Hmm.
Q
Exactly.
and then you run that and
Hmm.
then you can basically compare the difference in the protein structure, like the first one will have no beta turns and the
Hmm.
second one will have beta turns, so in that way the protein structure has changed.
Exactly. Yes. Perfect. Yeah. Thank you so much, Akrit. Now, can anybody tell me in Uniprot which uh, did we go? I mean, which link did we use to actually look at the difference in the variation? Like what effect? Yes, Sanjit, you can go ahead.
Yeah, after we entered Uniprot, we first went to
Yeah.
variant viewer, but then we saw a huge number
Hmm.
of variations in the protein, so
Exactly.
we kind of skipped that variation, and then we went to feature viewer, where
Exactly.
we located
And
the DNA,
hmm.
I mean the position of the mutation, and
Hmm.
then and then at the right side and the left side we selected the sites as the DNA binding. We got it that it's
Exactly.
a problem in the DNA binding site, and
Yeah.
then we copied the Foster sequence and we
Hmm.
proceeded with our other.
Yes. Perfect. Yeah. Right. Yeah, Revki, go ahead.
So when we are considering that, we found that the gene we're considering, that particular position in that protein sequence
Hmm.
lies in the DNA binding part. So
Yes.
that is why it's something of significance. If it weren't lying, then what
Hmm.
would be our inference?
So you can conclude that okay, since it is not in the DNA binding site, it may not affect the protein's function much. Okay, so that is what we'll be concluding because essentially your P53 protein needs to bind with DNA, and then only it will be able to arrest the replication, so that other cells does not proliferate quickly. So if it is not in the DNA binding, then we can assume that okay, yeah, maybe this mutation is not that problematic. It may not really affect its function. Okay, so we can come to that conclusion. But again, we need to verify it with other websites also. Like Finva needs to say, okay, yeah, it is not a problem, something of that sort. You need to understand that, and then yes, you'll be done.
Oh, okay. And if you know what is the graph below, like that red and gray graph, what does that show exactly?
See, actually, all of these, I am not very sure of this because they're very complex graphs, and I'm also learning only very recently. So this ones I'm not exactly very sure of it, but next time once I go through all of it, I'll let you guys know. Is that okay?
Yeah, yeah. Thanks.
Okay, okay, cool. So yeah, guys. So this is exactly what we did today. And can anyone tell me what we use the CleanMart database for? Like, what is the purpose of using CleanMart database? Like, what does it show? Can anyone tell me? Anyone, guys?
So,
Yes, exactly. Hmm.
ClinVar, it is a web tool under NCBI. It gives us
Hmm.
information about different diseases and the pathogen which is responsible for causing a disease, and it also includes
Yes.
mutations on gene that give rise to a disease, and it
Hmm.
also sometimes tell us the significant effects of drugs.
Yes, exactly. Yeah, perfect. Yeah. So CleanMart database is actually to show you, yes, whether there is if a particular mutation is pathogenic or not. Uh, if how diseases are caused, I mean diseases, what change is actually happening at the genetic or protein level, and also to see what the effect of drugs are there on our body. So all of this information we can acquire it from the ClinVar database. So you are absolutely right. So I think yeah, that sums up, that pretty much sums up what and all we covered today. Okay. So just will stop presenting now. Okay, guys. So that was it for today's session. Do you guys have any other doubts? Was it all clear today, or did anyone feel like it was a bit too much, or anything of that sort? Was it all good, guys? All right, that's great. And you have been a perfectly interactive crowd today. It was amazing. I love this session. Do you guys want to have more such sessions, or do you feel like this is a bit boring? We need to have something different. Do you guys like this sort of session? Okay, that's great. Awesome, guys. Okay, so in our upcoming sessions, we'll try to bring in such case studies more often, and we'll try to increase the complexity step by step so that it is not too much for you guys to take in. And I'm sure, like as we go ahead, you'll be able to use a lot more tools together, and we'll be like today, we'll be introducing little by little new stuff. Like today, I introduced you to ClinBar. Next time, we'll go for two new tools, three new tools. We'll increase the complexity, and we'll try to have much more fruitful sessions like this. Okay, guys. I'll just stop recording.